Hi there, my name's Joseph Williamson. I'm a final year PhD student at Queen Mary University of London, and I'm here today to talk to you about our new research that's just come out in the Journal of Applied Ecology on how riparian buffers can act as microclimatic refugia in oil palm landscapes. Now, a lot of you will have heard of oil palm, and those of you who haven't have probably heard of the product, which is palm oil. Now, palm oil is a vegetable oil. It's largely grown in Malaysia and Indonesia, and those of you who haven't consumed it in the last 24 hours may have used it in a cosmetic or some kind of cleaning detergent. In order to grow oil palm, you have to convert existing lowland tropical rainforest into oil palm plantations. Now, this poses a threat alongside anthropogenic climate change to terrestrial biodiversity, such as this charismatic megafauna here, but also to more functional groups, such as the invertebrates. Now, what are riparian buffers? Riparian buffers are tracts of non-production habitat that are retained in agricultural landscapes when you convert them from natural landscapes. You can see an example of one here in the top left with forest running through the middle of a very young oil palm plantation in Malaysia. And now this is primarily done to protect hydrological processes by preventing soil erosion and agricultural runoff. But more recently, we've started to view riparian buffers as habitat fragments in their own right. So a series of studies across a variety of taxa have shown that riparian buffers do support terrestrial biodiversity, including orangutans, jaguars, dung beetles, and dragonflies, although not all in the same landscape. We're also interested in how they might provide ecosystem services, both within them and in terms of spillover into oil palm and other agricultural types. And finally, we're also interested in how they might connect up fragmented landscapes as habitat corridors. This is shown to be the case for dung beetles, birds, and moths. But what we don't know anything about is the microclimate of these riparian buffers. Now, why would we want to know about that at all? Well, microclimate, or climate more broadly, is known to drive species development, physiology, and behavior, and through those, it will also drive species distributions. But it's often been predicted at poor resolution and with low accuracy. But this is rapidly changing as new technologies come out. A good example of this is a paper by Tommaso Jucca at Bristol, who used data logger data in combination with airborne LiDAR data to predict temperatures across a tropical landscape at a 50-meter squared resolution. Microclimatic data loggers are also being used by many groups now. So Stephen Hardwick showed that oil palm, which is in grey here, in the middle of the day is much hotter than logged forest in blue or old growth forest in red. But what we don't know is where riparian buffers would sit on this diagram. And what we definitely don't know is how the temperature you find within riparian buffers would then go on to affect the biodiversity they're able to preserve. So to start investigating some of these questions, we went to Malaysian Borneo in Southeast Asia, to the SAFE project, which is a large-scale fragmentation experiment, meaning big bits of forest have been retained in oil palm plantation when it's been converted. But they don't just have big fragments. The SAFE project also has riparian buffers of variable widths, meaning that it's an ideal place for us to go and study microclimate. So Matt Strubig, Eleanor Slade, and Sarah Luke were behind the design of this project, and Harry Heroin was the lead research assistant. And what they did is deploy 300 data loggers across the SAFE project in riparian habitats. There were 20 rivers were studied with 3 transects on each river, 5 loggers on each of those transects, which I promise adds up to 300. Now, these data loggers are essentially USB sticks that measure temperature and humidity every 30 minutes for 2 to 7 weeks is how long they're out in the field for, with a handy sun shield in the form of a polystyrene plate. Now, this is an aerial view of a riparian buffer, and this is what one of the transects roughly look like. We have loggers in the oil palm plantation here, and then we have them at a variety of distances from the oil palm buffer edge within the riparian buffer. So what we found in terms of temperature here first, so we've got maximum temperature on the left, this is daily, and mean temperature on the right, is that continuous forest, which is in both diagrams in blue, is much cooler than oil palm. As I've already said, this is like well known. But what we didn't know is these riparian buffers where they'd sit in the middle, and you've got the riparian buffer core, which means not close to the riparian buffer edge, so more than 10 meters away, is very cool compared to oil palm, or largely cool. A lot of them are even overlapping with values of continuous forest. Now, the riparian buffer edges were significantly hotter, sometimes even than oil palm, but to learn more about that, you'll have to read the paper. So we've shown that riparian buffers are kind of an intermediate temperature to continuous forest and oil palm with some overlap with continuous forest. And the same is reflected in the humidity data. This is actually VPD, which is almost the inverse of humidity. So you can think of it as dryness, and you've got the same patterns as temperature going on in maximum and mean daily values. Now, what we wanted to do from there is to go on and look at what's driving these microclimates. What is it that means that one buffer is hotter than another? Well, to do this, we use airborne LiDAR scans. Now, airborne LiDAR consists of flying a plane over a landscape and using a laser mounted on it to scan in the vegetation and topography. Now, if you're Tom Swinfield or David Coombs, what you can do with those laser scans is generate big maps of the landscape, looking at, so here this map is the canopy height across the landscape, and just to show you the resolution of this thing, you can actually see individual crowns and branches, but they also generate a variety of LiDAR metrics from these scans. This is just one of them. And what we can then do is map this up with our data loggers and see what is driving the microclimate that they recorded. So on the left here, we have plant area index, which is just a measure of kind of vegetation complexity. And we've got mean temperature on the y-axis. You can see with these results very nicely that as the vegetation gets more complex, you get this decrease in the mean temperatures, probably because of increased shading and decreased exposure in general. With topographic position index, you've got a positive relationship. So high topographic position index scores mean you're on a ridge. Low means you're in a depression. So this is to do with the topography. And what we're finding here is that in these depressions, you've got lower temperatures. On top of the ridges, you've got hotter temperatures. Again, this is to do with exposure, probably. You're going to have more exposure to sunlight on the top of ridges. And finally, we looked at maximum canopy height, and we got a very clear negative relationship between the height of the canopy and the temperature you find in the understory. Now, this is 3 metrics out of a whole host of ones we looked at. And also we looked at different microclimatic variables as well, but you're going to have to look at the paper to find out more results on these. These are just indicative of some of them. What we were also really interested in is the width of these riparian buffers. Now, this is something that is, buffer widths are mandated often by law. So in Malaysia, it's a 20-meter buffer, but it varies around the world. Now, what we wanted to know is if you increase this 20-meter buffer, what are the impacts that this has on microclimate? And what you can see is on the y-axis, we've got maximum temperature on the left and mean temperature on the right. And you've got this, again, this decay function of maximum temperature with the distance from the buffer edge, which you can just think about as the width of the riparian buffer. Now in green at the bottom, you've got our continuous forest controls. So we can see that at around the 80 to 100 meter mark, riparian buffers are starting to look extremely comparable to continuous forest in terms of their microclimate. What's interesting is this is the kind of region where other people have been saying this is the width you need to conserve certain biodiversity. And this is just to show these patterns again are mirrored in the humidity data. But what we want to know is how this all comes together to affect biodiversity. And to do that, we chose an indicator group, the best indicator group, dung beetles. Now, this is Proagadurus watanabe, a particularly fine specimen with big major horns that you find across our study landscape. Now, Eleanor Slade has been studying the dung beetles of SAFE for at least 8 years. And using human feces baited pitfall traps, she's deployed 6 major trapping campaigns, generating abundance data for the entire community across the whole landscape. Now, for this study, we took 2 of those campaigns that were focused in riparian buffers and generated diversity metrics for the dung beetles found there. Now, when we match this diversity data up with the microclimate data, we can start to see what's driving these diversity patterns in the landscape. What we find here, we've got maximum temperature on the left, mean temperature on the right, but instead they're going along the x-axis this time. We've got diversity running up the y-axis. Now, if you focus on the left diagram for the moment, we've got this black line here is the response slope of dung beetle diversity to temperature at a set buffer width of 20 meters. So this is quite a narrow width. And what we've got is this negative relationship whereby dung beetle diversity is crashing with increased temperature. As you increase that buffer width, this slope levels out. So at 80 meters, you've got basically a flat slope, meaning that temperature is no longer driving decreases in diversity. And this is mirrored again in the mean temperature, and also in a lot of the humidity data shows very similar results. And the important takeaway of this is that both buffer width, i.e. the size of your fragment essentially, and the temperature are driving the diversity patterns within riparian buffers. And they're synergistically interacting when they're narrow and hot to really hammer the terrestrial biodiversity. Now, what we need to know is how this impacts policy, because that's how we change things. The first thing we want to say from our results is that if you want to protect microclimate and also biodiversity, dung beetles, but also everything that they indicate, they should be wider. They are currently in Malaysia, they're 20 meters. We're saying more like 80 meters would be great. We also showed that vegetation quality is really important for protecting the microclimate within these riparian buffers. So we've got to start thinking about the vegetation quality there. And we've got to prevent practices such as logging or grazing happening in these buffers, and potentially even replant them with forest species to restore them. Topography is also really important factor to consider when designing these human modified landscapes. And in general, we need to just be thinking about microclimate when we're thinking about managing them, especially in the face of anthropogenic climate change. Now, if you want to know more, you should go and read the paper. It's in Applied Ecology, or you can follow me on Twitter. I would like to thank all of these incredible people, a lot of them who did a lot of the work behind this, and a lot of them who helped me with the aspects of the work that I did, and all the institutions that funded this project and made it feasible. I'd also like to thank the research assistants of the SAFE project, who don't get mentioned enough. These 3 prime suspects being Anis, Noi, and Didi, but there are many, many others. They just didn't have quite as good selfies with me. These are some references. If you have any questions at all, I think we have questions after this video. So hopefully you can ask them to me. If we don't, then send me an email, or if you don't want to, please send me an email. I'm really excited to discuss this with anyone. Thank you so much. I look forward to speaking to some of you face to face.